Hi, my name is Drew, and I'm here to briefly describe the Hershey and Chase bacteriophage experiment for you. This is for the prompt 31B. As society reveled over Avery, McLeod, and McCarty's findings on how DNA was the transforming material as opposed to proteins or RNA in 1944, more research began on the nature and purpose of DNA. Two researchers by the names Hershey and Chase set out to discover how DNA containing bacteriophages infected other cells. Bacteriophages are a type of virus that consists of two parts, a protein capsule and DNA or RNA genetic material. It was unclear at the time which part entered a cell and infected it. The answer would prove to be necessary in future attempts at deterring infections of viruses through the stalling of these processes. On the left, you can see a detailed illustration of a bacteriophage attacking an unsuspecting cell. Here is a simplified overview of the experiment Hershey and Chase conducted using my obviously expert art skills. Here we have two bacteriophages, one with sulfur labeled on the protein capsule and the other with phosphorus labeled on the DNA within the capsule. On the left side, we can see that as the bacteriophage infects an E. coli cell, the sulfur-containing capsule is left on the outside while DNA is injected in. After Hershey and Chase isolated the coats of the bacteriophages, no radioactivity was present, indicating that the protein had not been transmitted to the cell. On the right side, DNA is marked with phosphorus and tracked as it infects more E. coli cells. Once isolation is complete, infected bacteria appear, indicating that DNA is part of the bacteriophage that is injected into the cells. In conclusion, radioactivity detected on the specimens where the DNA had been labeled with phosphorus indicated that this was, in fact, the material passed on to the phage progeny and the infected cells. Lastly, here are my references, and thanks for watching.